Okay, so let's get started. So the first one, so we have Youngjae.
Hello.
Please come in front of the podium here, because otherwise people can't see you.
Hello. I'm Youngjae Park from South Korea, and I don't want to be a first presenter, but I'm a first presenter. So here in today's session, I prepare a very noise efficient chopper stabilized amplifier with large electric DC offset cancellation and very high input impedance. So our target application is monitoring the biopotential signals such as EEG or EOG or ECGs. And we think that our system can be applied into various biomedical systems such as depth of anesthesia monitoring or biomedical wearable devices. So to achieve large electro DC offset cancellation and very high input impedance, I propose dual mode DC subgroup and dual positive feedback to boost the input impedance. And yeah, thank you so much.
Okay, so Alicia is next.
Hi, I'm Alicia. I'm a final year PhD student at UC Berkeley, and my poster is about a neural prosthetic with human in the loop shared control. So my poster consists of 4 different categories that we all pulled together for this system. And the first is a novel brain-inspired HTC computing paradigm, which I'll introduce in my poster, and then how we achieve really low power in-sensor biosignal classification with this algorithm. Then taking that paradigm and seeing what we can do in the robotic space for robotic navigation. And then finally tying these things all together for a shared control feedback system with EMG-based prosthetic. So if you want more information on how we do this, please visit my poster.
Hi, I'm Vamshi. I'm a 4th year undergrad at Berkeley.
I'm Jen. I'm also a 4th year undergrad at Berkeley. Current bioelectric prostheses utilize 2 sensors, one on the abductor and one on the adductor, limiting the user to 2 degrees of freedom. Our device gives us 20 degrees of freedom by using 64 electrodes that can individually map to muscle groups.
So to sort of design a control paradigm, we're using the same brain-inspired hyperdimensional computing and taking data from a very high dimensional EMG electrode array and using a new paradigm called HTC recall to enable control of our robotic hand.
Okay, Pumia, yes.
Hi, I'm Pumiao Yen. I'm an electrical engineering PhD candidate from Boris Merman's group at Stanford University. I'm here to present our work, which is part of the Stanford Artificial Retina Project. And the current trend in brain-machine interfaces is to record from increasing number of neurons and to build towards the next generation of neural interfaces that has the capability of simultaneously recording from, like, say, tens of thousands of channels. Our group proposed to tackle the bandwidth, wiring, and power challenge with a compressive wired or analog to digital conversion readout architecture. While we achieve over 100-fold of lossy data compression comparing to like general Nyquist sampling, we demonstrate that we're still capable of capturing over 80% of the spikes and for potential action potential, and also like preserving the firing rates and enough information for further on, such as like standard spike sorting. And so there we're showing that WiredOR is a promising architecture for future neural interfaces. Please check that.
Okay, so now we have Cortec people joining us.
Yeah, so hi everyone, my name is Alexis and I'm a neurobiologist and got my PhD from the Microsystems Engineering Department at the University of Freiburg. And at Cortec, I'm the preclinical study lead. And of course, if you want to go to humans, we need to test everything in animals before. And that's our main development product here, the brain interchange, which we tested in several preclinical studies in sheep. And I want to show you with this poster that you can do actual research with it. And it would be nice to just demonstrate this to you at my poster. Thanks all.
Martin.
Hi, I'm Martin, CTO of Cortec. At Cortec, we have developed a technology for making implantable electrode arrays. And it's in our case, it's not about new materials, it's about old materials, only the traditional implant materials that we process with lasers and build electrodes for animal research, but also for clinical research. And we got FDA approval for a couple of those products that we developed. And this poster is about how we apply it to spinal cord stimulation electrode, but could be really anything. So, thank you. And there's brochures. I have brochures at my poster.
Hello everyone, my name is Mehtab Ramazani and I'm a third year PhD student at UCSD EC department. So I'm here to present our recent work, which is called eCannula, which is the integration of transparent graphene electrodes with imaging cannula to study hippocampus. And we simultaneously recorded 2-photon calcium imaging and also echoic signals at the surface of CA1 region. And please stop by my poster to talk more in details about my work.
Doing really well, Madison. Yes.
Oh, yes. Hello, I'm Madison Wilson. I'm a PhD student here at UC San Diego in the ECE department. So our work is on organoids, which, as Dr. Mora Lopez mentioned earlier, are three-dimensional clusters of stem cells that can be used to model complex organs such as the brain. And one potential application of organoids is in neuroprosthetics, where if you lose a function or damage some brain region, organoids can be generated from the patient's stem cells and then implanted to restore that brain function. So what we did was we implanted organoids in the mouse brain, and using our transparent microelectrode arrays alongside imaging, we found that organoids did in fact form functional connections to the visual cortex of the mouse and started to exhibit similar electrophysiological responses. Our work is the first ever demonstration of organoids connecting to a sensory network in vivo, and our technology offers a unique platform for studying these organoids as neuroprosthetics. So I look forward to seeing some of you at my talk or my poster.
Bye.
Hi, everyone. My name is Yong Wang, and I'm presenting on behalf of my student, Daifang. Basically, we're interested in epilepsy. So it's a severe neurological disorder, and patients' quality of life can be, severely impacted by this disorder. The good news is that about 70% of the epilepsy patients can actually lead normal lives, with anti-epileptic drug treatments. But the rest of them, who do not respond to at least 2 AED drugs, AED treatment, which might need surgical resection of the part of their brain that's considered diagnosed to be capable of generating seizures. So in partnership with Enigma Epilepsy Initiative, we have begun to look at the brain structural patterns that correspond to favorable surgical outcomes. So, if you're interested in some of the pilot exploratory approaches that we've kind of conducted, please visit our poster. Thank you.
Jancy.
Hello, everyone. My name is Janqi Zhang, and I'm a second-year PhD student in UCSD. And here I'm presenting our work using an unsupervised approach to understand human behaviors and in the context of bipolar disorder. And we all know that animal behavior has been well studied in the field, but an open field human paradigm has been missing. So here we're using an unsupervised approach to codify the pose and using an autoencoder to automatically understand what is the behavior difference between the control versus our bipolar patients. And we found significant different features in their pose in terms of their behaviors. And if you're interested in our work, please visit our poster. Thank you.
Hi, I'm Jonathan. I'm a second year master's student in bioengineering here at UCSD under Dr. Nodri and Dr. Callenbergs. We propose a frequency modulation EXG-based solution for addressing improving ABR testing for pediatric hearing loss, specifically to address some of the issues with ABR test quality, such as motion artifacts and hardware limitations, which lead to a longer testing time and discomfort for pediatric patients. Specifically, our FM approach is hardware and DSP based, and our demodulation schema provides improved computational complexity and performance over prior work. And with our high SNR ability, we propose that, or we hypothesize that we can overall improve the ABR testing modality and provide resilience to motion artifacts and reduce the overall testing time for pediatric patients. Thank you.
Hello, my name is Masaki Nakanishi from UC San Diego. So in this clinical work, we are developing a moving visual stimulus to assess visual dysfunction in the afferent and different pathways in multiple sclerosis. So investigations show the visual dysfunction in both afferent and different pathways, but there is no practical way to assess them simultaneously and conveniently. And we designed the moving stimulus, so which is flashing and also moving around the screen. So it is supposed to fixate the stimulus, and then we can use visually in both EEG potentials and also eye movement. And then I'm going to show you, today I'm going to show you, present a preliminary result. And then we are also developing a portable device like this, integrated with a VR headset and also a dry EEG sensor. So if you're interested in this work, please come to my poster.
Hello, I'm Jung Woo from Department of Biomedical Engineering, Gaming University, Daegu, South Korea. I will present part of my study. It's in my poster. We are going to introduce a BCI-based neurofeedback system, which could give feedbacks according to users' engagement while performing their motor exercise. Particularly, we are going to show you in this poster the effect of the motor cortex activities according to presence or non-presence, absence of virtual avatar's hands, showing first-person perspective in virtual reality. If you are interested, please come and see our poster for more details. I hope to see you in front of my posters and have some discussions. Thank you.
Hi, my name is Sensao. So the purpose of this study is to construct neurobehavioral model of complex motor skill. So our main tech is juggling tech. So we have observed the brain during juggling with EEG signals. So yeah, so as a part of the result in this poster session, we found how the brain recognizes ball movements. So please stop by my poster if you're interested in juggling. Thank you.
Hey guys, I'm Cynthia. I'm a second year PhD neuroscience student here at UCSD. I'm going to be presenting about VR-induced fatigue. You're probably wondering, what does the hangover have to do with anything in this conference? But this has in common, if you play VR a lot or you're just a VR fanatic, there's this thing called VR fatigue or VR hangover. And essentially what it is, other than motion sickness, there's a lot of visual and cognitive fatigue associated with it. And just in general, why do I care? Why should we care? Well, essentially this phenomenon is very important because I work with clinical populations. This is one of the #1 complaints I get whenever we do VR studies. So I'm just trying to find neural signatures that are involved within fatigue. So if you want to know more or just talk about the hangover, stop by my poster.
Hi, I'm Ryan Cave. I'm from Professor Knowledge Group at UC Berkeley, and I'm here to present a poster on performing drowsiness detection with user-generic EEG. Some key highlights of the work are the manufacturing process that we use to make our electrodes, which is a 3D printed based electrolysis plating process that allows us to make any arbitrary shape and leverage different types of materials. And we've used this ERP system. The other key highlight is the actual drowsiness detection that we performed across 9 users and across a couple different training and validations. If you're curious, please come by the poster and I'll talk your ear off.
Hi folks, I'm Srini. I'm a first year PhD student at CogSci at UCSC. And if you work on certain kinds of decoding models for certain neuroprosthetics, you might be interested in stopping by the poster. Why? Because I'm trying to work on some of the 3 main challenges in this field, which is limited neural data availability, limited subject availability, which is a good thing overall, but it's bad for neural engineering, and recalibration, which is constantly needed. So the principal idea is to kind of bring in data sets from different other domains, from non-neural data, from healthy subjects into this. So yeah, feel free to stop by.
David. Hi, I'm David DiFilippo from the Department of Music here at UCSD. So we're doing a synchronization study of a musical improvised duet and analyzing EEG data through recurrence analysis to see if there's any shared mental states kind of in the long run, but just sort of looking at recurrences in a shared phase space. Yeah, looking at interpersonal coordination areas and auditory perception areas. Thanks. And you have 2 minutes, right? You got 2 minutes.
And yeah, this is a coupled oscillator system I've been developing for sound synthesis that exhibits this phenomena called chaotic itineracy. So it was originally designed as an interactive real-time computer music instrument, and it produces an attractor landscape with chaotic transitions that kind of function like a search. Chaotic itineracy can also be used to model intentional sequences of behavior, default mode network, mind wandering, and sensory expectations.
All right, we made it. So lunch is served outside.